All right. Hello, everyone, and welcome to today's CryoEM Current Practices webinar. You're joining the 3 national centers established by the NIH Common Fund Transformative High-Resolution Cryo-Electron Microscopy Program. My name is Christina Zimani. I'm a scientist and training liaison at the National Center for CryoEM Access and Training, NCCAT. I'm joined here today by my colleague here at NCCAT, Ed Eng, as well as Michael Schmidt from the Stanford SLAC CryoEM Center, S2C2, and Lauren Vega from the Pacific Northwest CryoEM Center, PNCC. NCCAT has invited today's speaker, Helen Donelick, and I will introduce her properly after my colleagues from each of the centers give a quick overview and update on the CryoEM resources that we offer at no cost to the research community. This webinar is a regular event that we host usually the last Thursday of every month at this same time, but we make an exception in November and December to avoid the busy holiday weeks at the end of those months. We'll be meeting on December 9th, and then in January again at the end of the month. Each month, our talks highlight the methods that researchers are using to obtain and interpret the data they can collect at the national centers. And we are recording today's talk. You'll be able to find this recording on our YouTube page or the events page at cryoemcenters.org. And on that site, you can also find registration links for future talks and general information about the larger NIH Common Fund CryoEM program sites. So you can actually go to that site and already register for our December 9th talk. PNCC will be hosting Tarjani Thakur from the Tomasic Lab at the University of Arizona. And her talk is titled Determining Structural Mechanisms of Allosteric Regulation in ABCC Family Transporters Using a Remote CryoEM Pipeline. So save the date now for that talk. You can already go register if you'd like to join us in December. A couple of final logistics. Please use the Q&A feature to send in questions at any point during the talk, or you can also upvote other questions you see there. If you have questions directed to logistics or access at the national centers, our panelists here will respond to those directly in the Q&A box. I will be moderating questions for Helen. If there are some detailed questions that might be best to answer on the spot, I may interrupt her, but otherwise we will save questions for the end of the talk. With that, I'm going to turn over to our center representatives, and I'm going to let Lauren go first.
Awesome. Thank you, Christina. So my name is Lauren Vega, and I'm the project coordinator for Pacific Northwest CryoEM Center. So we currently offer one proposal type, both for single particle analysis and tomography, giving up to 480 hours a year for 2 years. We do have a monthly submission deadline for applications on the first of each month, and each one of our proposals is delegated what we call a SPOC, which is a scientific point of contact, in order to discuss your scientific questions. We have 5 scopes. So we have 1 Arctica with K3, a Krios with Falcon 3 K3, 2 Krios with Falcon 3 BioQuantum K3, and 1 Krios with Falcon 4 BioContinuum K3. For sample preparation, we have a VitraBot, a Leica GP2, and a VitraJet, which is still undergoing kind of validation before we offer that to users. We're in beta testing. We're also under restrictions still for COVID, but we're offering one-on-one remote training and small remote workshops. Once we can have visitors on site again, we do plan to offer those small on-site workshops. So once we can have those, we're very happy to have people back on site. If you're interested in PNCC, apply today.
Thank you, Lauren. And I will turn over to Mike.
Thanks, Christina. I'm Mike Schmidt. I'm one of the co-directors of the Stanford SLAC CryoEM Center, S2C2. And we have much of the same equipment. We have 3 Krios microscopes full-time for CSC work. And we also have time on another Krios and a Thales Arctica especially for training purposes. We're especially proud of being able to do both remote and limited on-site training here. We have currently more than 10 people who are being trained either remotely or on-site for more or less local people. But please feel free to contact us, especially for the training and for the use of the microscopes too. The website is there. Thank you.
Thank you, Mike. And I will let Ed take over next.
Oh, welcome. We're here in New York City, and we have three main proposal types at NCCAT. One is high-end instrumentation access on one of our 4 dedicated Krios instruments. We offer an embedded cross-training program where trainees can spend anywhere from 1 week to a semester here to learn CryoEM technology. And we have a new category called grid preparation and screening. All users are welcome and encouraged to come on site for any of these access categories. Something that's coming up this March, we're going to bring back our single particle short course. If you want to find out more, November 18th, the NCCAT user office will have an info session. We'll run it more like an AMA session. If you want to sign up, email the NCCAT user office at nyspc.org or find out more on our website, nccat.nyspc.org.
Awesome. Thank you, Ed. All right, and now I have the privilege of introducing today's speaker, Helen Donelick. Helen received her bachelor's degree at Pacific University in Oregon and is now a PhD candidate at the University of Utah, co-advised by Brenda Bass and Peter Shen. When Helen is not in the lab, you might find her playing trumpet in concert band. But today she's going to be telling us about RNA biochemistry and how she's using CryoEM in her thesis research. So thank you, Helen, for joining us today, and we're looking forward to hearing about your work.
Thank you, Christina, for that introduction. I am very excited and very honored to be here today with all of you to discuss some of my work for my thesis project. So I think we can all appreciate that it is an RNA world out there, especially today. With COVID being a real problem for the past almost 2 years, this is an RNA virus. So how do we as organisms fight these type of viruses so we can go on and see to live another day? Very simplistically, these viruses that are RNA viruses have a viral double-stranded RNA intermediate, where these double-stranded RNAs can be targeted by double-stranded RNA binding proteins. This will then elicit an immune response, leading to the destruction of the virus. Now, in the system that I study, which is in Drosophila or fruit flies, the antiviral pathway is called RNA interference or RNAi. Now, RNAi may sound familiar to you, as RNAi is commonly utilized in the biological fields to be able to study gene knockdown, the use in genome-wide RNAi libraries, and even use in therapeutic applications, such as viral infections, cancer, and neurological disease. Now, while RNA interference is commonly used, we're still learning more and more to this day about the mechanisms behind this pathway. So to give you a general idea of RNA interference in Drosophila, I'm going to walk you through the pathway, where viral double-stranded RNAs are targeted and processed by dicer2 into small interfering RNAs or siRNAs. Additionally, this essential protein dicer2 can also process endogenous or self-double-stranded RNAs with the help of an accessory cofactor called LOXPD. This leads to the formation of endogenous siRNAs. In either case, these siRNAs have to be loaded onto the RNA-induced silencing complex, or RISC, to be able to target the downstream RNA. And this is done through the formation of another complex that includes dicer2, the siRNA, and a cofactor called R2D2. Here, one strand of the siRNA, called the guide, will be loaded onto Argonaute2, where dicer2, R2D2, and the passenger strand are no longer needed, leaving Argonaute2 and the guide to make up active RISC that can now target the viral transcript for degradation or mRNA for silencing. Now I'm going to direct you to dicer2 for a moment to talk about the biology and the structural information we know about this protein. Dicer2 is a unique enzyme in which it's able to process double-stranded RNA depending on the termini or the end of that RNA. This first video I'm going to show you is how dicer processes blunt double-stranded RNA, which represents viral-like. Here in a moment, we'll see this viral double-stranded RNA come into the picture, and it's going to bind to the helicase domain. The helicase domain will then clamp down onto this double-stranded RNA. We'll start to see it unwind and thread through the helicase domain, being cleaved by the RNase-3 domain in the middle of the protein. And you can see that there's a flashing icon in the helicase domain, and this is indicating ATP hydrolysis. On the other side, 3' overhang, which is representing your self-like self-RNA, this will bind directly to the Topaz platform domain at the top of the protein, and then will be cleaved by the RNase-3 domain into exact 22 nucleotide products in an ATP-independent reaction. We also have some structural information regarding the protein dicer2. Here, I'm showing you ApoDicer2 solved to 8.7 angstroms. I also noted the molecular weight of dicer2 at 197 kilodaltons, just to show you that dicer is on the smaller end of proteins that are studied by a CryoEM. But overall, we can see that this protein is L-shaped and it has 10 subdomains. Most of them are labeled here. We can also see that the helicase domain is in an open conformation in the absence of double-stranded RNA and ATP. But whenever we add double-stranded RNA, in this case, it's specifically blunt with ATP gamma S, we can see that the double-stranded RNA is partially unwound in the helicase domain, which was revolutionary to the field. We can also see that the helicase domain is in a closed conformation clamped down onto that double-stranded RNA. Now, while these structures were revolutionary and very informative, you can see that they are in general lower resolution structures. And pointing your attention to the dicer2 helicase domain, even though the full-length protein was used for these grids, we're only able to see the helicase domain, which means we're missing over half of the information of this protein. And with ATP and double-stranded RNA, we have the hypothesis that this means, for one, we know it's a functional helicase domain, but it likely adds to the overall dynamics of the protein, making it very flexible and difficult to study by CryoEM. Fruit flies are not the only organisms that have dicer proteins. Many organisms have dicers, including us. And this structure I'm showing here is human dicer bound to its accessory cofactor TRBP. Human dicer is in brown and TRBP is in green, bound to its pre-let7 RNA. And this structure was solved to 5.7 angstroms. And this is a higher resolution structure than what I just showed you before for Drosophila dicer2, but this RNA is not in the active site near the RNase-3 domain, so it's not cleavage competent. So we're not getting information on the mechanisms of how the protein cleaves the double-stranded RNA. Additionally, a very recent paper that just came out in Science in 2021 of dicer-like protein 3 bound to its pre-siRNA. Here we actually have a cleavage competent structure where the RNA is pushed up against the RNase-3 domain, which will make the cut on the double-stranded RNA, and the double-stranded RNA binding domains are pushing the RNA into that active cleavage site. But this protein also has a helicase domain, which is not modeled here due to high flexibility of that domain. So we're not getting cleavage competence and the helicase domain at the same time. And this is a recurring theme with dicer proteins. And the final structure I'll show here is, again, a very recent structure that just came out in Nature Plants for Arabidopsis dicer-like protein 1 bound to a primary microRNA. This time, we can see the double-stranded RNA in the active site, so it's cleavage competent, and we have the helicase domain. But at 4.6 angstroms, it's not high enough resolution that you can get residue information to understand exactly how the protein residues are interacting with the double-stranded RNA. So you can see overall, there's major themes that are coming up with studying dicer-related proteins and current hurdles when using CryoEM. Dicer proteins are quite flexible and dynamic, and dicer proteins can also exhibit preferred orientations. When dicer proteins are on the grid, they prefer to lie on their side, so it looks like the letter L, and we don't get as many orientations of other views. Also, obtaining a cleavage competent full-length dicer structure is challenging. You either get cleavage competence or you get the helicase domain. And if you get both, it's low resolution. So one of my goals for my thesis project is to obtain a cleavage-competent full-length structure of dicer with the helicase domain at a high resolution. So to be able to try to get to that idea, we decided to avoid, for starters, studying just dicer with double-stranded RNA, instead focus on another region of the RNA interference pathway, focusing on the RISC loading complex, which includes dicer2, siRNA, and R2D2. And since I gave you some background to the dicer2 protein, I want to do the same for R2D2 as well. Now, probably whenever you hear the word R2D2, you're thinking of the droid from Star Wars that's small yet very powerful. And the protein R2D2 is actually not that different from this droid. R2D2 is a small double-stranded RNA binding protein at 35 kilodaltons, and it has 2 double-stranded RNA binding domains. And R2D2 is very important for RNAi. In this example, I have Drosophila embryos that are expressing GFP in wild-type embryos, and you can see when you introduce GFP double-stranded RNA, the GFP signal goes down, indicating that RNAi is active. But if you mutate R2D2, in this case, R2D2 is knocked out, there is now no difference between the presence or absence of GFP double-stranded RNA. Both embryos are still expressing GFP, indicating that RNAi is now inactive. Now, while I'm not showing it on this slide, R2D2 is not required for siRNA biogenesis, but is instead required for moving the siRNA onto Argonaute2. But R2D2 also appears to act as a switch, allowing dicer2 to have multiple functions. And this can be seen through dicer2 on its own processing a very short double-stranded RNA at 25 base pairs, where we can see an additional siRNA product is produced. siRNA products are generally 21 to 23 nucleotides, so a very similar size to this double-stranded RNA. But if you add dicer2 and R2D2, we can now see that the amount of additional siRNA product is greatly reduced. So R2D2 prevents the processing of short double-stranded RNAs, essentially telling dicer to say, nope, you're no longer processing this RNA, we need to move it on to the next step. Now, besides R2D2 having a great name and the numerous Star Wars memes we can make, why should we study this protein? As I mentioned, R2D2 acts as a switch, allowing the essential protein dicer2 to have multiple functions in RNAi. But how this complex forms and passes the guide strand onto Argonaute2 is unclear, even though this is an essential step in RNA interference. And also, because we're structural biologists, we're curious if adding a cofactor could help stabilize dicer2, so a higher resolution structure can be solved of dicer2 bound to a cofactor and a double-stranded RNA substrate. So to be able to do so, we have to purify these proteins. And it would be an ideal world if we could just express dicer2 on its own and R2D2 on its own and combine them together and freeze grids. But unfortunately, R2D2 is actually unstable without dicer2. Here, I'm showing a Western blot of a knockdown of dicer2, which you can clearly see on the Western blot. But we also can see that the R2D2 band is greatly reduced. We've additionally attempted to purify R2D2 on its own in the BAS lab and have been unsuccessful. So to overcome this issue, we express dicer2 and R2D2 at the same time using the baculovirus expression system in SF9 or insect cells. And here we specifically only tag dicer2 with an OSF or a 1-strep flag tag and leave R2D2 untagged. That way, any R2D2 that we get back, we know was bound to dicer2. And this can be seen through whenever we purify dicer2 and R2D2 through affinity and size exclusion chromatography, where we get a clean dicer2 band and R2D2 band with very minimal contamination from any other proteins. Now, before I move into my CryoEM work, which is the reason we have these webinars, I want to just briefly cover some biochemistry that I did with this complex. So I incubated my dicer2 R2D2 complex with a radio-labeled siRNA to study the binding affinity. And what I hope you can appreciate by looking at the quantification on the left and the raw image on the right is that siRNA binds to dicer2 and R2D2 with a high affinity, calculated to 4 nanomolar, or estimated to 4 nanomolar through the binding assays, and in an ATP independent manner. And I did test dicer-2 on its own in a very similar experiment, and it binds to the radiolabeled siRNA at approximately 1 to 2 micromolar, nearly 1,000-fold weaker than that of the complex. And R2D2 is not stable on its own, so it wouldn't be binding siRNA on its own. So we're fairly confident this KD reported is specific to the dicer-2 R2D2 complex. So with this information, we felt comfortable with moving on to CryoEM. So let's throw this complex on CryoEM grids. And here, again, I'm just showing you that I purified my complex through Finney and Sky's exclusion chromatography, where this fraction was then used for the CryoEM grids, where we see a clean dicer-2 band and R2D2 band with very minimal contamination of other proteins. I then create a reaction mixture of dicer-2 and R2D2 with an equal amount of siRNA in the presence or absence of ATP. Now, while I just showed you on the previous slide that ATP was not required to bind the siRNA to the complex, we are still curious if there would be any conformational changes in the helicase domain upon the addition of ATP. And then I have my freezing conditions below, which we froze our grids using the Vitrobot. And I will note that we'll use quantifoil R22 grids to be able to use multi-shot recording. We then collected data sets at both the University of Utah Titan Krios, which is where I'll spend a majority of my time talking about that data set. And also we collected a data set at the Simmons Electron Microscopy Center with the New York Center for Structural Biology, which I'll briefly mention today. So first, I just wanted to show you one of my micrographs that I got from this data set. And again, I have my freezing conditions to the side. And probably if you're an experienced cryo-EM scientist or maybe new to it, you're probably looking at this micrograph and going, that's not the best micrograph I've seen in my lifetime. And it's definitely not the best micrograph, but we decided to collect a data set off of this anyway, because we were curious of what we'd be able to get from this kind of data. So I'll briefly walk you through our data processing workflow, where we had collected a little over 2,000 movies because the micrographs didn't look stellar when we initially collected them. And they went through pre-processing with motion correction and CTF estimation. I then used a blob-based picker to be able to get 1.4 million particles. And we were relieved to see that after 2D classification, we are able to see the traditional L-shaped 2D classes of dicer, indicating that even though at the micrograph level, it didn't look great, we were still able to get useful information from that data set. These 2D classes were then used for template-based picking, and where my data workflow starts to diverge slightly from traditional methods is I use both template-based picking and also the neural network of method picking called Topaz, which after template-based and Topaz picking, I was able to get slightly under 600,000 particles that then went under 2D classification again, where the best 2D classes had 240,000 particles. I then did 3D classification on those best particles and got 133,000 particles for the best class that underwent heterogeneous refinement to our final 6 angstrom structure that we have today. Now I mentioned Topaz, this neural network method of picking. And for those of you who might not be familiar with Topaz, Topaz is a more newly developed technique to pick particles that are present within your data set, but just aren't being found by traditional template or blob-based picking methods. And you can see that before Topaz, for my dicer-2 R2D2 complex, I had an overrepresented amount of side views and not as much with any other orientation. So Alex Noble gave a virtual talk at the University of Utah back in December of 2020. And I was actually driving home in the middle of a blizzard to visit my family for the holidays. And I thought, what a better time, not while I was in the car, but when I actually got to my parents' house to try Topaz, because I wasn't near my bench, I didn't have experiments to run, and it was a great time to utilize the computational tools that I had at my disposal, especially whenever we were looking at some of my views. And this is an example helicase domain where I was getting very few particles, only a few hundred, and the resolution was very low. It was very even difficult to tell if this was the helicase domain. So after learning how to use Topaz in several iterations, I was able to not only get now several thousand particles, a little over 8,000 here, and the quality of those 2D classes greatly improved, where we're able to see the traditional C-shaped helicase domain with an additional density on the helicase domain that we've now attributed to R2D2. And we can even start to see some secondary structure within this class as well, which we had not previously seen before in the helicase 2D classifications. And after Topaz, we can see our distribution map here. Even though we still do have an overrepresented amount of certain views, it is greatly improved as how it had been in the past. And just I'm going to walk through different ways of how Topaz improved my data set, both at the 2D classification level, particle distribution, and the 3D classification level. But let's first look at the 2D class level. So before Topaz, you can see for the side view or the traditional L shape of Dicer, it was fantastic. And we did not have to use Topaz on those views because we can see secondary structure within them, and they were really high quality 2D classes. But you can see before Topaz, they were the majority of the particles that I was getting at 87%. And after Topaz, I will note that I downsampled the number of side views because they were still overrepresenting my data. So it's now down to 66%, which explains why after Topaz, I have less particles than I had before Topaz. With the helicase domain, again, we have very low quality, very poor 2D classifications, where we can kind of see an extra density on the helicase domain, and it's marginally C-shaped, but only 4% of the particles for the helicase domain were represented. But after Topaz, it's now up to 15%, and many of the classes greatly improved, where we can clearly tell it's the helicase domain with an additional density coming off of the side. For the front on view, and this is if Dicer is laying on its side and you flip it towards you, so the front of the L is now coming out of your screen. Before Topaz, we weren't even sure if this was actually the class or not. And it was only 0.4% of the total particles. But after Topaz, it is now 16%. And we can see it's improved, but it's not high quality. It's still low resolution. And because of this, we've actually denoted this class as the gummy bear class because it looks like a gummy bear. And other classes, which I'm not showing what they look like by 2D class, that represented 8% of the total particles before Topaz. And this is now down to 2% after Topaz. So maybe some of those particles are being better classified into different regions. We can also see the improvement of particle distribution before and after Topaz, where before Topaz, we can clearly see hotspots of particle distribution in views. And after Topaz, although we still have those hotspots, we can see that we're starting to fill in the blanks in between those regions, giving an overall better particle distribution map. And also now looking at the 3D classification level, we can see that before Topaz, for one, I had a total reconstruction of 9 angstroms, where at the helicase level, we see a very large extra density coming off of the helicase domain, which we first attributed, oh, that's likely R2D2 in the siRNA, but it seemed too large overall, and it was a little bit spiky. But after Topaz, we can see that the overall resolution improved for the total protein at 6 angstroms, but the helicase view looks much cleaner as well. And we still see that extra density on the helicase domain, and the overall size of the extra density makes more sense for the size of R2D2 because R2D2 is quite small. And finally, looking at the front on view of Dicer before and after Topaz. Before Topaz, this view was underrepresented and it looks very streaky. It's almost like if someone had a wall of wet paint and ran their hand through it, that's what my model looked like. And it's very spiky as well. But after Topaz, that streakiness has greatly been improved, but it's still low resolution. We can also see there's this extra density coming off of the RNase-3 domain. And at this time, we're still not sure what that extra density is. We're planning on collecting, of course, additional data sets for the Dicer2 R2D2 complex, hopefully achieving higher resolutions overall with all the different views to understand better what this extra density could be. Now, Topaz was very informative and very helpful for finding underrepresented views within my data set, but I still have a 6 angstrom or a lower resolution structure. But what can we actually learn from my reconstruction at this time? So this is my reconstruction with just different side views shown, and I fit my reconstruction with apodicer 2 at 7.1 angstroms. And what we can see as I boxed in red is there's an additional density on the helicase domain that's not taken into account by apodicer 2. And when we initially saw these results, we thought, oh, that extra density is very likely R2D2, but possibly the siRNA as well, because the siRNA was included in this grid condition. But I'll show you on the next slide the reason we don't think this extra density also includes siRNA. We think it's specific to R2D2. So the reconstruction I'm now showing here was collected at the New York Center for Structural Biology, and it is overall lower resolution at 7.6 angstroms. But this grid condition had dicer 2 and R2D2 only, no siRNA, no ATP. And the concentration of dicer 2 and R2D2 was the same on the grids as I showed on the previous slide. But we see the same result where there's an extra density on the helicase domain that's not being taken into account by apodicer 2. And because siRNA wasn't present in this grid condition and we're seeing the same result as we did on the previous slide, we feel more comfortable at this time attributing that extra density to R2D2 rather than R2D2 and the siRNA. Now getting a little bit more specific into my reconstruction, I'm going to show you this short video here where I've color-coded the helicase domain with its specific subdomains. The helicase domain has a total of 4 subdomains. And I've pointed out where the extra density is as well. So while dicer's rotating on its axis, you can see that the extra density on the helicase domain is most closely associated with the orange density or HEL2I. And whenever we saw that result, we were actually really excited because we know it's similar to another dicer protein and its accessory cofactor. So again, I'm just going to briefly show you the reconstruction of apodicer 2 fit into the reconstruction, where we see that extra density associated with the orange or HEL2I subdomain. And now I'm going to show you how human dicer and Drosophila dicer 2 are very similar in domain organization. And human dicer has a very similar fold to dicer 2. I'm also showing you that the accessory cofactor for human dicer, called TRBP, is very similar in domain organization to R2D2 as well, and they're approximately the same size. And TRBP is also a double-stranded RNA binding protein, so it's expected to fold in a very similar manner as to R2D2. So with that information, we fit my reconstruction with human dicer and TRBP. And we can see that human dicer fits very nicely into my overall reconstruction, as we would expect. And we can also see that the third double-stranded RNA binding motif of TRBP fits nicely into the part of the density that we've attributed to R2D2. I will note that there is still extra density that's not being taken into account by either protein, but the authors of the human dicer TRBP structure were only able to reconstruct the third double-stranded RNA binding motif of TRBP because of the flexibility of the protein. So it's possible that I'm seeing more of R2D2 than they saw of TRBP, but my reconstruction is still low resolution, so potentially with higher resolution information, we'll be able to better understand what that extra density that I'm currently seeing actually entails. So as I start to wrap up my talk, I just want to give you some additional ideas that we are considering for my project. One is the use of pegylation of proteins before grid preparation. It was recently shown in 2021 that polyethylene glycol helps prevent aggregation and denaturation during the freezing process. And dicer really likes to aggregate and become denatured whenever you're freezing grids. So we're hopeful that this will help us avoid some of those problems. We also are exploring alternative cross-linking methods for the sample. One is glutaraldehyde. I have successfully cross-linked dicer 2 and R2D2 using glutaraldehyde. I just haven't put that sample onto grids at this time. And finally, we're still trying to find a way to get the siRNA into the dicer 2 R2D2 CryoEM complex. And one idea that we have is to purify dicer 2 R2D2 and the siRNA by size exclusion chromatography. So we can take specific fractions that have all three components rather than just the protein. And this way, we also avoid having to spike in the siRNA and wait a certain amount of time for the complex to form. And then we also don't have to worry about finding the ideal ratio of protein to RNA that will actually form the complex that we want. I wanted to give you just some take-home ideas before I completely wrap up my talk, where the Dicer2 R2D2 project, we continue to improve the resolution of Dicer2 proteins. The first structures that I showed you in this talk were solved at 8.7 and 6.8 angstroms, and now we're at 6 angstroms. Even though that isn't a significant leap, we're still confident we'll be able to reach a high resolution structure within the near future. I've also shown you that the R2D2 density interacts with the subdomain HEL2I, and that's similar to TRBP to human dicer. We're really excited about exploring the idea further if TRBP and R2D2 modulate their respective dicers in a similar manner. And finally, we're still working on obtaining a cleavage competent structure with the helicase domain at a high resolution. I've only discussed one of my projects today, but this is an ongoing project in the lab that we're excited to explore and to continue working on. And finally, if you're not an RNA biologist or someone that studies RNA interference, I wanted to give you some take-home ideas in general of what you can take away from my talk and potentially apply to your own projects. And the first is the use of Topaz. You saw that I spent many slides focusing on Topaz and how I was able to help my project find underrepresented views that were present in my data set, but just not being found by traditional methods. I think at this time, Topaz is a bit underutilized, but I've encouraged my colleagues at the University of Utah and specifically the members in my lab to try Topaz, and many have been successful. So I highly encourage anyone on this talk that's processing their data set and thinking, man, I really wish I had more views of this one side of my protein. Topaz might be able to help you out. Also, low resolution information can still be informative. The structure that I showed you today was at 6 angstroms. We are still able to get some biological relevance from that structure. But of course, we want the high resolution structure to get even more information. And with the many new advancements and techniques coming out in CryoEM, we're really excited and enthusiastic to be able to try all those ideas to get a high resolution structure, hopefully in the near future. Finally, I'll just finish with my acknowledgements. I'm a member of 2 labs, the Bass Lab and the Shen Lab, and I have to specifically point out my amazing mentors, Brenda Bass and Peter Shen. Without their mentoring, I definitely wouldn't have the opportunity today to be speaking to all of you. The EM core at the University of Utah, run by David Belknap and Dave Tim, not only for running a fantastic core, but for being mentors to myself as well. The DNA and RNA synthesis core at the University of Utah for making our RNA substrates. Our funding resources and the CryoEM centers, I'll specifically point out New York Structural Biology Center because they did do a data collection for us. But of course, S2C2 and PNCC for holding these webinars to be able to give more information to people that are learning CryoEM or interested in CryoEM, and for inviting me today. Thank you for your attention, and I'd be happy to take any questions.
Thank you, Helen. That was really fantastic. I didn't even get a chance to interrupt you. So if anyone has questions, please go ahead and send those in. There are a couple, so we'll get started with one. I'm going to take the privilege I get with having the microphone and ask the first one, actually. So your co-expression in SF9 cells, do you infect with 2 different viruses, or do you put both genes in the same baculovirus?
Yeah, that's a good question. For my Dicer2 and R2D2, I infect my cells with 2 separate viruses, but other members of my lab have actually now used a P-big vector, and they're able to combine multiple proteins within one, so they don't have to do the multiple viral expression. But I use 2 viruses.
Thank you. There's a question about your blotting conditions. What is your reasoning for doing a 20-second wait time before blotting?
So that was through multiple trials and error why we chose 20-second blotting. I was using 30 in the past, but then we weren't getting as good of ice quality. And I also went down even to low as 10. That wasn't ideal as well. 20 is just what we converged on that worked.
Great, thanks. All right, the next question, a little bit of background. So you showed that dicer plus R2D2 binds double-stranded siRNAs with an affinity of 4 nanomolar and dicer alone at 1 micromolar. Does R2D2 help with binding depending on the siRNA overhangs, or does it always just strengthen the affinity?
Yeah, that's a really good question. I haven't performed binding assays of longer double-stranded RNAs with dicer2 and R2D2. That would be something really cool to explore. And at this time, we're really not sure how R2D2 is actually interacting with that siRNA. I wouldn't be surprised if it's somehow interacting with the 3' overhang ends, but at this time, we just don't have enough information to be able to say for sure.
All right, thanks. Then does your structure have a uniform estimated resolution, or even at low resolution, do you find that there are regions that are significantly better than others?
Yeah, so I was trying to run local resolution before I presented this, but I just ran out of time. But if I lower the threshold whenever I'm looking in Chimera, there are regions that have higher resolution, specifically in the RNase 3 domains. That seems to be pretty stable, but the rest of the protein is still fairly flexible.
And yeah, I guess a natural follow-up to that, have you tried any focused refinement or multibody refinement?
Yeah, that's something I'm interested in exploring, is trying the multibody refinement or additional refinements after that.
Awesome. All right. And then there is a question that I also had. Did you try to fit an AlphaFold prediction of R2D2 into your extra density?
Yeah, that's good. I'm glad someone asked about AlphaFold. I did run R2D2 through AlphaFold, and that's another reason we were excited about comparing it to TRBP, is because it looked very similar in AlphaFold to TRBP. We actually even saw a third double-stranded RNA binding motif on the C-terminal end, which previously was expected to just be unstructured in R2D2. So I have attempted to fit those into the extra density, and it fits very similar to TRBP.
It looks like there's a question that was in the chat, and I don't know if it's been transferred to the Q&A, but a question was, I was wondering if you tried processing your older data using Topaz to see any improvement.
Yeah, I did. So I collected another data set of dicer2 and R2D2 back in August before I learned about Topaz. And I did process it there as well. I still didn't get a higher resolution structure. Overall, the ice was a bit thicker on that sample, so that inhibited us being able to achieve higher resolution, but it did improve the overall distributions that we were seeing.
And that plot you showed about the azimuthal plot of the orientations you found, that looks like it's CryoSpark. Was that all through? How did you do your processing, through CryoSpark or Relion, or a combination?
Yeah, most of it was done. All that I showed here today was done in CryoSpark. I have done analysis both in CryoSpark and moved those particles to Relion and did further processing, but it didn't greatly improve the resolution overall.
I had a question as well. So you said your sample where you include the siRNA, you did with and without ATP, but you only showed one reconstruction. So could you tell a difference between those conditions, or did they look similar?
Yeah, unfortunately, without ATP samples, we weren't able to collect a data set with, so we don't know if there's any conformational changes. But based on the fact that with ATP, it didn't look like there was a closed conformation of the helicase domain. We don't think there was any, but also we didn't see the siRNA there as well, so that might change upon the binding of the siRNA. Maybe then there'd be a conformational change.
Thank you. All right, another call for any questions. And give everyone just a minute to type if someone's frantically trying to get another question in there.
And if anyone has any additional questions, I'd be happy to answer them via email, or my Twitter is down below. I can also answer them there.
All right. If there are no more questions coming in, we can actually wrap up a few minutes early. I'll stall for another second or two. All right. So thank you, Helen, for that talk. We wish you well in your continued cryo-EM studies, and hopefully us at the center see some of your grids coming in soon. Thank you, everyone, for joining us today. We wish you all well in your cryo-EM endeavors, and the national centers are here to support your science. So let us know how we can help, and we will see you again here in December.